So our journey for the mRNA begins at the nucleus, where it comes out of the nucleus and is in the cytosol of the cell. It's free floating and it attaches to a free floating ribosome, where it will begin translating. Upon translation, you'll have a SRP attached to it and
attached to the signal sequence.
Correct. Attached to the signal sequence and halt translation of the protein. We will then move down to the SRP complex will move down to the SRP receptor, where it'll attach, and the SRP receptor will then bring it to the translocon, where SRP will then detach itself, and it will push the translating protein through the translocon, where translation will once again resume. Upon this, the translocon plug will detach.
From here, the ribosome will continue to translate the mRNA through the ER membrane and into the ER lumen. At this point, when it's still translating, the oligosaccharide attached to the dolichyl phosphate will be transferred over to the polypeptide by the oligosaccharyl transferase. It will then attach. The mRNA will continue to be translated into the polypeptide. Once it is completely translated, the signal peptidase will cleave off the signal sequence, and we are left with just a polypeptide. At this point, the oligosaccharide will be trimmed of 2 glucoses. Now at this point, this glycoprotein will move in the ER lumen over to the calnexin. We got to make sure that the translocon plug is reattached to the translocon and seal it up so nothing else can enter that protein channel. When the glucose of the glycoprotein is attached to the calnexin is a chaperone protein, which helps the protein fold properly. Once it is folded, the protein disulfide isomerase will add disulfide bonds to this polypeptide so that it will keep that 3D proper form. After this product is made and this folded glycoprotein is made, glycosidase will remove the final glucose of this oligosaccharide. At this point, the glucosyl transferase will check over this folded glycoprotein, and if improperly folded, the glucosyl transferase will add one more glucose, and it will return to the entire process and be folded again. However, if it is properly folded, which it is here, it will strip it of its final mannose and will continue to move within the ER lumen and attach to a transport vesicle. This transport vesicle will move along the cytoskeleton into the cis side of the Golgi. Once the glycoprotein enters the Golgi apparatus, enzymes within the Golgi will either add or remove sugars on the oligosaccharide and will modify the oligosaccharide into a different sugar. This glycoprotein will then move through the Golgi apparatus, and once again, it'll leave through the trans side this time, and once again be attached to a transport vesicle.
The example that we're using for this is the 6-mannose-phosphate, because we're going to the lysosome of the cell. The RIB will attach to the tethering protein, and what is essential here is that the RIB is specific to the tethering protein because it's the identification for the transport vesicle. The tethering protein pulls this transport vesicle closer, allowing for the V snare and T snare to attach and intertwine. Once they are able to attach and intertwine, they begin intertwining further, which will bring in the transport vesicle and cause fusion of this protein.
This fusion allows the glycoprotein to actually enter the lysosome.